Well, hello everybody. This is Brian Culley. I'm the CEO at Lineage Cell Therapeutics, and I'm very happy to be here presenting at NobleCon17. Lineage is a public company, and I may be making some forward-looking statements, so I refer everyone first to our Safe Harbor Clause, where you can learn more about our risk factors and go through our filings. All right, so first, Lineage Cell Therapeutics, from an overview, we have a very different approach. The company transplants in an off-the-shelf manner cells to treat various diseases and conditions. So there are diseases and conditions for which certain cell types in your body become dysfunctional or die off. So we make those cells and then we transplant them to the body. So it's a particular form of cell therapy. We call it allogeneic cell transplants. We control our own manufacturing. We have a 2-suite GMP facility, and we have one of the largest cell therapy patent portfolios with many hundreds of applications and issued claims. And we have 3 clinical stage programs. So we have something we call Oprogen, which is in a phase 1/2A clinical trial to treat dry age-related macular degeneration with GA, and I'll obviously talk more about that. We also have OPC1, which is an oligodendrocyte technology to treat spinal cord injury, and our VAC platform, our oncology platform, can generate a number of different projects or programs rather, but we are currently using it in a phase 1 trial in non-small cell lung cancer. One of the exciting things about cell therapy is that using whole cells can sometimes generate effects that seem to be beyond the reach of small molecules or antibodies. So for example, Lineage is the only company to actually report an ability to restore retinal tissue. So we transplanted retinal tissue into a patient and made an area of damage smaller. And that's never been reported before. It seems like it would not be possible to do that with a small molecule or a traditional approach, only through some sort of transplant. We also have seen very encouraging effects in our spinal cord program and our oncology program, and I'll get into that. All of these lead to billion-dollar commercial opportunities, and in many cases in areas where the competitive landscape is relatively light compared to others. So that's also an advantage for the company. In the near term, we have some exciting news. We're going to do a data update this quarter and next quarter with our dry AMD program, and we expect to complete enrollment in our non-small cell lung cancer clinical study. As of our last reported quarter, which was September 30th, we had about $38 million, and the company varies, but as of the end of the year, our market cap was about $264 million. So to get back to our base technology, we're really trying to pioneer a new branch of medicine, the transplant of whole cells. Now, this has been around in the form of a bone marrow transplant for many years, very successfully, but transplanting other cell types is really beginning to hit its stride. We do this because we start with undifferentiated stem cells. Now, we never put a stem cell into a patient. A stem cell for us is just starting material. It's just a place for us to multiply and scale a number of cells. We retain them in this undifferentiated state to a point, and then we begin to move them through a process where we direct their differentiation. And we're able to make, as I described, any of these cell types. There are over 200 cell types in your body. We have 3 of them in the clinic: the retina cells, the spinal cord cells, and dendritic cells, part of your immune system. The amazing thing is that these undifferentiated stem cells harbor the ability to become anything. They can become any cell type. If you have the recipe, you can make any cell type in the body. So we manufacture these 3 and are using those in the clinic, but in theory, we could go into many other areas as well. And by the way, we do this through a very natural process. We expose the cells to different compounds and chemicals and different durations and temperatures in order to direct their differentiation. We never change the DNA. We don't go in and manipulate the genome of these cells before we put them into the patient. And because they come from cell lines, these are frozen cell lines, we can scale them up to tremendous numbers, enough to satisfy any of the even largest disease indications which are out there. So we're not starting with a patient's own cells. We are taking these cells from cell lines that are well established and well characterized. All of our manufacturing is an area of excellence for the company. The things that we've been able to sort out and the scale we've been able to get into, I'll describe, and that's generated a lot of intellectual property as well. Going back to those 3 clinical stage programs, you can see them all here. What's quite notable is that each of these clinical programs has received some validation, some financial support, whether that's from the Israeli Innovation Authority or the California Stem Cell Agency or Cancer Research UK. You can see here about $40 million in cash and in-kind services to support these 3 clinical stage programs, which I feel reflects positively on their value. And they also are progressing well through early stage clinical trials and beginning to approach middle stage and later stage clinical studies. So beginning first with AMD, this is one of the leading causes of vision loss, and we manufacture RPE cells to address this condition. The hallmark of the disease shown in these 6 gray boxes is the progressive loss of retina cells. So as you see that black smudge that's getting larger over time, those are retina cells which are dying off. And in connection, following the red line, the patient's vision is becoming severely impaired. This individual in just a span of 7 years has gone from normal vision through legally blind and then beyond to be almost completely blind. And that's because those retina cells are dying off very rapidly. This is a massive problem, which also means it's a massive commercial opportunity. People are often familiar that wet AMD, which is a different form that we're going after, that wet AMD, those products sell about $10 billion a year, but there's nothing approved for dry AMD. It's kind of remarkable, and yet 8 to 9 times more people suffer from dry AMD. So we think there's a huge commercial opportunity, and specifically the group that we're going after, there are probably about 2 million people in the US that have the form of dry AMD that we think we could start with. Okay, so why? How can it be that you have this huge commercial opportunity and there's nothing approved? Well, we think traditional approaches don't work in this setting. We think that what you need to do is replace the whole cell. And that's because by the time a cell dies, it's not one little biological pathway that has gone wrong. The whole darn cell is, totally, confused and lost. So we think the way to fix that is to replace the whole cell. So in this cartoonish figure, you can see on the left side, those dark blackened areas, those are RPE cells. Those are the specialized retina cells which we manufacture. And they're dying off. They're dying off in connection with the disease process. And in doing so, the cells which they support become sick and fail. The waste material, which they ordinarily clear, that begins to accumulate, and you can see that in the eye. You just look in, you can see this yellow smudge or these yellow dots that accumulate, and these are some of the hallmarks of the disease. So what if you could manufacture brand new retina cells and transplant them to the eye? Could you rescue those photoreceptors? Could you clear that waste material out? We not only believe so, we've shown evidence of this. So in the clinical study, which we have recently completed enrollment, there are 24 individuals who finished this study. The first 12 of them, they were all legally blind. So that's a safety cohort. It's hard to show efficacy in people who are legally blind. This is not, going from zero vision to perfect vision. That it's overly ambitious to expect that from cell therapy. We're really trying to slow the disease or stop the disease, or in some cases make some clinically meaningful benefits for these patients. But we started with people who are legally blind because you're not going to do any harm to people who are who already have such poor vision. And so we were very excited. We saw a very nice safety profile. The cells were well tolerated. They remained in every patient for a long time, more than 4 years in the longest cases. And that allowed us to move into the second group of 12 patients. These are individuals who have better baseline. These are more like our customers. These are the people who have problems with their vision. They're suffering from dry AMD. They are headed to a bad place with respect to their visual ability or visual acuity, but they still have vision. I mean, some of these people could still drive a car, right? 20/65 would be the best vision in the second cohort. And this is where we're seeing the effects. This is where we're seeing our target patient population seems to be driving some nice clinical benefits. I mentioned already that the tolerance of the cells, I can tell you that in those 24 patients to date, we've had 0 cases of acute or delayed rejection. So the fears that people have that you put foreign material into the eye and it's going to be rejected, those fears in our hands have been unfounded. Every single patient has retained their grafts, and as I said, even as long as 4 years. We also have seen very encouraging early signs of efficacy. It's not every event in every patient, but we have evidence of a reduced growth of atrophy. better vision, better reading speed, the structure of the retina. I mean, the great thing about the eye, you can look inside and see what's going on. You can take these images and cross sections and aerial views of the retina, and in doing so, you're able to see what's going on back there. And there have been some signs like reduction in waste material and the structure of the retina that are very encouraging for us. And also that retinal tissue restoration, again, only company to show that that was possible in a human being. So what do people care about? People care about being able to see. People want to be able to see their phone. You lose your central vision. I mean, think about it. Your car, your phone, just even making eye contact with people, you lose all of that. So people care about vision. So we, of course, follow vision. And in this green line is a measurement of the numbers of letters that people can read on an eye chart with their treated eye and their untreated eye. So the green line is the treated eye, the orange line is the untreated eye. It's a nice control. Most people have two eyes. You treat one, you leave the other alone, you see what happens. And what we have seen is that people who receive our treatment seem to be reading about 10 or 12 letters longer, and it's durable. That's very encouraging. Now, these numbers are a little small. We don't have 100 people with this, but it's a very nice early indication. And this is in part why our next two data updates are going to be so important, because we have 6 more patients that were treated this fall, and they're going to be going through these early months, and we're going to be able to add a lot of data to this slide, which we presented last November. Do 10 or 12 letters matter? Sure. We have a very respected retinal advisor who said if you can show him 5 letters, you have his attention. And so here's what that looks like, 10 or 15 letters, the kind of changes that you can have. So you can imagine if you take this and translate it to a book or a monitor, what a huge benefit it is. This is exactly the kind of eye chart that you read when you're going for your driver's license. So think about the benefits that you could have, especially with elderly patients. That's really who this is most prominently affecting, are the elderly. The other thing that's going on is we measure a very important metric, which is the area of cell death. So these cells tend to die off starting in one place, and then it grows outward. You can think about this like lighting a match underneath a piece of paper. You're going to have a little bit of burn and a tiny dot, and it's going to expand outward. And so you have an inverse now of these curves. You can see the orange untreated eye. It's getting worse faster. The area of atrophy is growing quicker compared to the treated eye. Now, these are very subtle changes, just a millimeter or so. So it takes a long time to see these changes, but we think that as we continue to collect data, that these error bars will narrow and it'll be reinforced that we are having a positive outcome on the area of atrophy. This is probably the most exciting piece of data that we've ever reported. This is evidence of something that it really should not have happened in a human being. People cannot heal their own retinas. So if I have a cut on my arm, that's a different kind of cell, and those cells will heal over time. But if you have a problem with your retina and cells die off, you can't heal that on your own. And so these areas of atrophy only get larger. And you can see that. These are aerial views. We're out here in San Diego. We have wildfires, and so we get accustomed to looking at these wildfire maps, these aerial views of fires that only burn outward. And so we can do these tracings. And so this is an image of a person's retina, and it's traced and colored to help you see it. And you can see that the orange area is at -1 year. So this is a year to go from orange to red. So you can see that the orange area has burned, right? The cells have died off and it's gotten bigger. So we have confirmed that this person's area of atrophy is getting worse over time. Then we treated them. And then we looked back at 9 months. And now you fill in the area of red, which is baseline, and the area of blue, which is at +9 months. And the blue area was smaller. So that was striking, because you can't normally expect that. These are areas of atrophy that only get bigger. And then it's continued out to 23 months. It's still a little bit smaller than it was at baseline. We've taken this out to 3 experts in the field of retinal imaging. We've had them read it and affirm it. And these are not just aerial views. You can get, and we have cross sections, and you can see all the little layers of the retina, the ONL, all the different layers of the technical aspects of what's going on. And you can literally see that there is greater density and structure and organization to the areas which have recovered, which, by the way, overlap with where we've placed the cells. So it's really exciting. We think that we may be able to have a real game-changing differentiated approach here if we can continue to provide this kind of benefit in a greater number of patients. So what does that mean as I kind of lead into the commercial setting? Well, only cell therapy, as I explained at the outset, has the potential to restore tissue and to do so with infrequent dosing. So this isn't like wet AMD or some of these approaches where you got to poke the eye every month and stick a molecule in there and have patients, elderly, going into the clinic every month after month after month, and they don't feel any different, and all you're doing is slowing the disease process. The cell therapy approach is we expect to be a one-time treatment. And only Lineage has shown that maybe it's not enough to slow the area of atrophy. Maybe we can actually freeze it or reverse it. And we also are the only company that has access to a really exquisite delivery device that was originally created by Johnson & Johnson, is currently in the hands of a company called Gyroscope. And we have an exclusive relationship with Gyroscope to deliver cells to the subretinal space. I'll show you what that looks like, but the takeaway from this slide is that I think the company is very well positioned vis-a-vis the others that are working in this field. But we don't just rely on our cells, we rely on cell delivery too. So on the left cartoon here, this is the traditional way of delivering cells. You poke the eye, you stick the needle all the way back, you shove it under the retina, and you form a bleb. You deliver the cells into that area that's light blue or gray, called a bleb. That's fine until you take the needle out, and then all the cells come flying out of the hole that you made, and they go to other places, right? They're live cells. They can grow, and they can cause problems and lead to adverse events and complications. And this is a problem with the field. Everyone who puts cells in a subretinal space has this problem, perhaps until now. We use the Gyroscope Orbit SDS, which allows an encapsulated needle to go through a flexible cannula around a suprachoroidal space. So it's going around the back of the eye and up underneath the retina, as you can see in the right image. And when you extract the needle from this side, you don't have efflux of cells going into that vitreous cavity. So you avoid the problem. And the benefits here are not only around safety, but also around dose control, right? If you have total dose control, you have less variability in your clinical trials, and that means that you can have smaller and faster and cheaper clinical studies, potentially with more compelling evidence by having less variability. The left side, you have a lot of variability. Those, you don't know, what, 50% of your treatment blew out of the cell into the vitreous? I mean, that's a real big challenge. So we have, what we're trying to put together here are the best cells, the best delivery, and overall the best option for physicians. Further along that lines, we are preparing for success, and we are establishing commercial scale manufacturing capabilities. Cell therapy, a lot of people talk about the problem with scale, and reproducibility. We can already manufacture the equivalent of 2,500 clinical doses in a 3-liter bioreactor. So we can manufacture 5 billion RPE cells that are more than 99% pure. And we formulate them in an easy-to-use thaw and inject formulation. So these cells are frozen, and when they are put into a warm, like a water bath, and then they're put into a needle, it takes 5 minutes to get them all ready and get them into the injection needle and into the patient. And we're not doing this starting with the patient's own cells. These are cell lines. So we can easily scale those 3-liter bioreactors up to much larger bioreactors, or daisy chain them all together, and to go into 5-figure patient production and beyond. We already manufacture far more clinical trial material than we're ever going to use. So we think that this is an important differentiating feature of our program, and an area that we put a lot of effort in is the manufacturing. So that leads to a long list of bullets on this slide, because this is a list of how we've tried to position for commercial success. There are a lot of advantages here. I won't go through them, but suffice it to say that regulatory, commercial, clinical attributes here, protecting the intellectual property, there are a lot of ways that we think we're creating value here. All right, so that's the first of 3 programs. The other 2, I'm going to go a little bit quicker. The second one is very emotionally exciting. It's spinal cord injury. We have individuals who, sometimes their cars crash, and that leads to a paralysis. And what we do is we look to manufacture the cells that die off, the cells that are lost after a spinal cord injury. This happens to about 18,000 people a year, and it's very expensive to care for individuals who don't have mobility. And so our goal is to improve their quality of life and get them better mobility. Cervical injuries are usually graded by the nerves in your neck. So the higher, C1, C2, those nerves are responsible for things like respiration. When you get down much lower, you get out to, for example, your fingers, your control of your finger. If you could move someone, as in the blue circles, from a C4 to a C6, there's a tremendous amount of quality of life and tremendous cost saving for the system, moving someone from a C4 level of injury to a C6. And you can see that here, your ability to move a wheelchair, feed yourself, etc., is all enabled when you go from C4 to C6. So these are the kinds of things that we're trying to accomplish. And in fact, we have had about a third of the patients on the clinical study move to motor level units, C4 to C6, C5 to C7. This is what we've seen. And we do that by manufacturing the cells that have died off and transplanting them into the spinal cord. Imaging, it's really easy to kind of look at this image, this panel up above, prevention of cavitation. Cavitation is a hole. A hole is formed after the injury, after the diving board accident, after the mountain bike or motorcycle accident. The inflammation leads to cell death, and you can see that on the left. This is an animal, it's not a human slice, but this is an animal slice. But in that red circle, you have this cavity where the cells are absent. So you cannot carry an electrical impulse across a gap. Electricity doesn't jump a chasm like that. You have to provide some material. So we provide the material in the form of the cells that can myelinate, which means they can provide the electrical insulation to the nerve axons that allow you to have mobility. And so this is the same sort of thing we're looking to accomplish in the clinical setting. I won't go through, I'm just going to mention that we've had 25 patients that have been treated according to the schema, and what we've been really happy about is that 96% of them had a successful engraftment, so again, no rejection of this material even years later, and about a third of them had 2 or more levels of improvement in their mobility. So that also was quite exciting, and it's been very well tolerated. No serious adverse events have been related to our cells. In order to make that jump, as I said, a third of patients had that significant jump. Some patients do have spontaneous recovery. I don't want to suggest that this is only solely attributable to our treatment, but we think we are adding things. So we would imagine that patients would be doing electrostim, they'd be doing physical therapy, but they'd also get a dose of our cells to help give them some substrate from which they can build greater mobility. The overall clinical data certainly supports further evaluation in a randomized clinical trial, and that's something that we are planning on. But we had to fix some things first. We had to fix the manufacturing. We wanted to get the manufacturing in a much more professional and pre-commercial status, and that's something that we had worked on and just announced a couple of months ago. We have a new ready-to-inject formulation. We increased the scale tremendously. We brought in all sorts of new analytical methods. New intellectual property was created. It's been really exciting to watch the team take what is basically a research-grade process and introduce commercial attributes so that this program can get back into the clinic. And to give you an example of a before and after, these gray bars are measures of non-target or undesirable cell types in an old population. The company, before we got our hands on it, they were making a lot of, let's say, mixed populations of cells, and you can see as high as 20% undesirable cell types. But in orange, the Lineage material is really clean. We have very low levels of impure cell types or undesirable cell types, and we think that that's going to be very attractive to the agency and also beneficial to the patient. So we, again, are going to be preparing to head back into the clinic with this program. We have to talk to FDA and do some other things, but it's been a great breakthrough for us to be able to show that we could deploy our manufacturing, not just in retina cells, but also in these oligodendrocytes. And then we're going to move on to dendrocytes, or dendritic cells, and apply the same technology to our oncology platform. But we do something a little bit differently. We manufacture dendritic cells because they present antigens to your immune system. So your immune system needs information, sometimes external help, right? That's what a vaccine is. It's giving your body some external information and some help to know what to fight against. Well, the same thing can happen in oncology. Your tumor is growing, it's foreign, it's gone, bonkers, it's not normal tissue, and you want to identify it as such. And we can use dendritic cells and we can load them with antigens in order to educate your immune system that the tumor needs to be destroyed. And these dendritic cell approaches have had some success in the clinic, which is helpful, but we're doing it again from cell lines. We don't start with a patient's own cells. Economically, that's not really efficient, and we think that doing it with the allogeneic cell lines, although it won't address every patient possibly because of matching issues, it is a very economically appealing way to proceed. So we have a clinical study right now, but this is importantly just one of many potential programs that we could have. We could, the programs here are defined by the antigen. So you could have an ovarian cancer antigen that's different from a lung cancer antigen that's different from some infectious disease antigen. Every antigen that you might imagine could be loaded into these dendritic cells and create a new program. So we're looking to expand how we develop the VAC platform by going further beyond just what we're doing right now with a non-small cell lung cancer antigen. We're working on this trial. I hope that we can finish enrollment this quarter. This study is being run by Cancer Research UK, and they're in the UK, and they're having a lot of COVID troubles, so it's kind of tricky, but we do think it's possible that we will be able to finish enrollment this quarter, and that would be a nice conclusion to this study and generate additional data for us, which we could then announce this year. Which overall, we have quite a hefty amount of news coming this year on each of our programs, most notably the OpRegen study with the dry AMD data updates, but the other two programs also have some significant and informative events this year as well. So Lineage objective, we're trying to take cell therapy forward. It provided a lot of promises many years ago, but now we have the tools and the talent, I believe, to be able to make it a clinical reality. So our manufacturing, our data, our intellectual property, our cash on hand, I think really positions us very nicely to become a clear leader in this area of regenerative medicine. And if you're really excited and interested in what I've talked about, there's a whole bunch of information on our website, including from patients who actually participated in our clinical trials, talking about their experiences and how important even small changes to their clinical outcomes can be. So Ahu, thanks very much. I think I can stop sharing and come back to a Q&A session with you.
That sounds great, Brian. Thanks for the presentation. It was a great overview of the company. So I'll start with asking specific questions to the program, and after that I'll get into more of the technology and some general questions. So my first question will be on OpRegen. You mentioned the competitive landscape. We know there are a lot of cell therapies, gene therapies, small molecules, antibodies in the space. How do you see Lineage approach fitting into this landscape? And also, if it is approved, do you think there would be opportunity for multiple approvals in the field?
Yeah, I think, I'd love to be able to go through like each and every program, but to kind of bucket them. An advantage we have over gene therapy is that when you are replacing a gene, you need to know that the patient has a problem with that one gene, right? Gene therapy doesn't fix 20 problems simultaneously. So even though there may be 2 million people just in this country with dry AMD with GA, how many of them have the one problem that gene therapy has the one solution? Right? So you're not going to really address 2 million people with gene therapy. You have to screen. And I know one company in particular is screening 10,000 patients to find the patient for their clinical study. So what does that mean? Like, how much do you have to charge, and how are you going to find patients that have one problem for which you have one solution? Our approach is different because I don't care why those cells died off. It doesn't matter whether it was pathway 1, pathway 2, or pathway 3. We replace the cell. Just like if I can't start my car, I don't care if it's the starter or it's a flat tire or I don't know. I just need to get somewhere. So give me a new car. That's our approach, and I think that we're well positioned with gene therapy. On the other side of it, with small molecules, it's kind of similar. Small molecules tend to have more breadth, right? You might be able to hit in a different pathway that has sub-pathways, and so you might get a little bit more bang for your buck or a little bit more horsepower. But again, you still have a certain amount of selection. So the question for small molecules is not so much are they helpful, but will they show enough of a benefit to be clinically relevant to the patient, and can you show that convincingly in a clinical trial that you can actually afford, right? So they have to have enough horsepower to be able to deliver a big benefit, even with nothing approved yet. So my view of that is that I think we're really well positioned through our transplant approach. If a bunch of approaches get approved, right? Let's give everybody the benefit of the doubt. Everyone has a successful few years, and there's a whole bunch of stuff on the market. Again, gene therapy, I don't worry so much because we're also probably one and done treatment. So, whatever piece of the market that gene therapy may have, that's okay. It might not be really noticed. On the small molecule side, most of those companies that are working in complement, which is a leading approach, they are working to slow the growth of atrophy. So they don't get rid of patients, right? It's like slowing the growth of a tumor. You still have a tumor. It's still going to kill you. In this case, the atrophy is still going to rob you of your vision. So I think that even if those other approaches are successful, that we can come in and basically be used. I think we could steal patients from them more easily than they could steal patients from us. Or maybe there's combination approaches. But really, I think the power of cell therapy, as soon as we figure out exactly how and where best to use it, I think it might be able to leave others in the dust.
That's quite helpful, Brian. So my next question will be, you mentioned you'll have some data updates on the Origin trial. What are we expecting in the first half of 2021? Will we see any further data on the retinal restoration patients?
Yeah, I hope so. So there's a few things that we expect to announce in the first half of this year. We have always been very forthcoming with the data, not just pooled data, but even individual level data. So we had 6 patients that were treated in the fall from September to November. They're now a few months in. So our plan is this quarter to provide an update on how they are doing at 3 months. 3 months is a little early, but it might give us some indication. It could be positive. It could be neutral. Even no matter where it is, I'm not going to be very conclusive, but I'm going to be looking for signs of some directionality from those 6 patients. Then there'll be an even more useful update when those patients are out to 6 months. That might be around the ARVO Medical Conference, which I believe is in May this year. Then the other thing that we're going to be looking for is retinal restoration. So when it took us 9 months before we even became aware of that retinal restoration patient, and then we spent a meaningful amount of time really making sure it was real before we went out publicly with it. And one of the things that was special about that patient was they got very aggressive placement of cells. So we have then gone back to the investigators and said, we'd like you to try and mimic that. We'd like you to be more aggressive with your placement of cells. So we have accomplished that in most of the patients that were treated this fall. So as they also get out toward 3 months, 6 months, and ultimately 9 months, we're going to be looking much more carefully for that retinal restoration. If we see it, we're going to want to share that with people, because that could be incredibly exciting. And we don't know when it could happen, but we believe that we have treated these patients more recently in a way that it gives us a great chance to be able to see more retinal restoration.
That sounds great. We will look forward to the data update for that program. So my last question on that program will be on the regulatory approval. What do you envision a regulatory approval would look like for you for this Origin program?
Yeah, the real answer will come after we see more data. So you want to pursue a regulatory endpoint that your approach can deliver, right? We have a new approach and there's nothing been approved. So there's a little bit of flexibility there to negotiate and discuss what makes sense. If we see an overwhelming signal in visual acuity, we would want to lean into visual acuity. I mean, that would, again, be very powerful. That's what people care about, is their ability to see. And I spoke recently with a woman who was on our clinical study, and she called it life-changing because she thinks she's going to pass her driver's license test, and she's the only driver in her household. So visual acuity is one path we could go on. But visual acuity can be subjective. It can be a little messy. You saw the error bars on that bar chart. It's not everybody's favorite endpoint, reading letters. So the other thing that most companies, most sponsors think is suitable with the FDA is the growth of atrophy. How fast is that area of atrophy growing, and can you slow it down? It's attractive. It might be even more achievable for us, but it is, again, an imperfect measurement. It takes a long time to see those changes. And we don't know today if our cells are better at one or the other. That data will come to us. But there's a third option that I think only Lineage has available to it, which is retinal restoration. You could imagine that we could design a clinical study looking at a responders analysis. What % of patients have an area of atrophy that is 10% larger or less? That would be quite powerful, because in a control arm, you're expecting everyone to get worse. These GAs grow about 1.5 millimeters square per year. So if we are looking at a control arm where everyone's doing worse, and we have a small number of patients who basically the disease is frozen or even reversed, that's a lot of statistical power that could help us be successful. So, those are 3 avenues, and we'll continue to explore them as our data continues to read out.
That makes sense. My next question will be on the VAC platform. So I am curious, what do you think about the potential of VAC2? And also, do you think you can use that platform for various indications in oncology?
Yeah, there's an interesting thing about VAC2, because VAC2 is both a proof of principle and also a product candidate, right? So in terms of a proof of principle, we're looking to demonstrate that an allogeneic cell line with an antigen can lead to high levels of reactive T cells, like specific reactive T cells. We want to interrogate the patient's immune system and ask if their immune system has noticed and responded to and amplified our specific message. And we have seen that in our interim data, and we hope to see reinforcement of that in our future data. So from a proof of principle aspect, that's where VAC2 can be very powerful. We're generating clinical data to demonstrate that the mechanism is valid. And then we could go back into all these other programs that prophetically some of these we haven't even designed yet. With respect to VAC2 as a product candidate, it also can be a product candidate in non-small cell lung cancer. That will be a larger question around what is the data, how have these patients done over time, what is the clinical data, and because not all of that data is available to us, it's still very preliminary. It's a little ambitious to say that we're definitely going to go do a large phase 2b in non-small cell lung cancer right now. We need to think this through and collect a little bit more data. But the nice thing about the VAC2 program is it does serve 2 purposes. It's a product candidate in and of itself, and it also sets some mechanistic evidence for other product candidates that we might imagine or that we might generate through alliances with other companies.
That makes sense. And my other question about spinal cord injury program. We see companies' presence in oncology, we see it in ophthalmology, and this is a different approach. Why are you pursuing spinal cord injury? Why does it make sense to use cell therapies?
That's a really good question. I mean, the kind of pithy short answer is we acquired this one. But why would we acquire it? Why would we spend money to acquire this program? And I think a big part of the answer is we have a fixed cost in manufacturing, and there's a great efficiency in having our manufacturing team work on multiple programs because they're trained and skilled and can apply their knowledge and intellectual property to it. But there's something different. There's something bigger. And that is that cell therapy, when it was first sort of conceived of, came out of the gate really ambitious, looking to treat autism and Alzheimer's, and maybe someday it will. But there's a little disappointment in the field that it didn't lead immediately to big successes. It has led to successes. I mean, you look at what's been accomplished with the money that's come from the California Stem Cell Agency. We see, amazing changes in individual cases like the Bubble Boy story and things like that. But I think the problem with cell therapy is it tried to do too much too soon. We didn't have all the tools. And what you need to do is go where it can really show that it's most powerful, right? Cell transplant, allogeneic transplant, where is that best applied? Well, places where you don't have a strong immune response, like the eye, like the spinal cord, right? Because then you're going to have better safety profile. You're going to reduce rejection. You've got a lower risk of, funky things growing that don't belong there. So, it's kind of the idea of lower hanging fruit, right? Go where you can be successful. And longer term, do I have the ambition that Lineage is going to have, a dozen different things and, making pancreatic cells and making different kinds of immune cells? Sure, like that makes sense. But you got to get some wins under your belt and show people that this really is a new branch of medicine that's going to be extremely big. And so going after spinal cord, I think, gives us that opportunity.
Thank you so much, Brian. We came to an end to our presentation, so much appreciate your time and participation to our panel. That was a great conversation. Thank you.
Great. Thanks so much, Dr. Demir. Appreciate it.